Good morning, everyone. I am Sadhana from the Department of Pharmaceutical Engineering. On today's class, I'll discuss about biotransformation of drugs in BPPK, the subject name Biopharmaceutics and Pharmacokinetics. So, what is biotransformation? So, in the pharmacokinetics, if we take drug into oral route, so first initially the drug will enter into the body, and it is enter into the blood, which we called it as absorption. In the pharmacokinetics, absorption, distribution, metabolism and elimination, we call it as ADME. In this, the absorption is the first step. So in this step, after taking the drug orally, it is entered into the blood circulation by the process of absorption. So after absorption, the drug distributed throughout the body, and at the site of action, it will show the mechanism of action, and it is enter into the liver. So here the major metabolism takes place. The biotransformation is also called as metabolism, where the active form of drug is converted into inactive form and it is eliminated through urine. So here the metabolism and the excretion. In the ADME, ME is meant for metabolism and E is for excretion. So both are combinedly called as elimination. So the elimination is a very important step after the drug showing the mechanism of action. If it was not eliminated through the body, it is accumulate and it produces the toxic effects. So that's why after absorption and distribution, in this unit we'll study about the metabolism and biotransformation of the drugs. So what is the biotransformation? So it is an enzymatic conversion of the active form of the drug to inactive form. So why we need to convert the drug from active to inactive? So when the drug shows the mechanism of action, later it is entered into the circulation and it is excreted through urine via kidneys. If the drug is in active form, most of the drugs in the biopharmaceutics are lipophilic drugs. So these drugs, when enter into the kidney, so it will not filter, it will reabsorb to the body, and again it is entered into the blood circulation and again it accumulates in the body, so which leads to toxicity. So that's why in the biotransformation process, we'll convert the lipid soluble drugs to polar and hydrophilic drugs. Thereby it will not reabsorb from the kidney, it is eliminated throughout the body. So here the metabolism is the essential pharmacokinetic process which convert the lipid soluble drugs and non-polar drugs to hydrophilic and polar molecules and excreted by different process. In that majorly urine is the main excretion part. So remaining by sweat, by any other by nasal, by rectal route, the drug will eliminate through the body. So this is because only the water soluble drugs can undergo the excretion process. See here the drug with different enzymes which is converted into a metabolite. So here in case of prodrugs, the drug initially is in the inactive form. Later on, after metabolism, it is converted into active form. So the biotransformation, it is specific, the chemical transformation for each and every molecule which is entering into the body from external environment. So the molecules which are entering into the body from the outside, we called it as xenobiotics. So these xenobiotics, which are the chemical substances present outside the body, say for example pesticides on the fruits and in the environmental air, so including oxygen, carbon dioxide, nitrogen, different types of chemicals, carbon monoxide, pollutants, everything are present in the air. So all these substances when entered into the body, we called it as xenobiotics. These are the endogenous compounds. So these xenobiotics means plant constituents, drugs, pesticides, what are the cosmetics we are using on our skin, and flowering agents and fragrances which we are used in the form of perfumes and food additives. So each and everything we consider as xenobiotic. So these xenobiotics are the substances, whether it may produce the mechanism of action, means any good effect or bad effect in our body, which are enter into the liver and excreted and biotransformed, and the inactive form of all these compounds excreted through urine. So here, see, the xenobiotics are different categories, which are highly lipophilic and lipophilic, polar and hydrophilic. If the substances are polar and hydrophilic, so we don't have any problem, because if the substance is polar hydrophilic, it is after metabolism, it is directly entered into the kidney and it is filtered out in the inactive form. But if the drug substance or any xenobiotic is in highly lipophilic and lipophilic form, so if it is highly lipophilic, so in phase 1 biotransformation, it is converted into polar by adding any hydrophilic moiety like OH, COOH group, etc. So in the phase 1 biotransformation, the highly hydrophilic molecules are converted into polar molecules. So these polar molecules and these lipophilic molecules, so both are undergoes to the phase 1 metabolism, and these molecules are converted into hydrophilic. So with this, next, these hydrophilic molecules and the polar molecules, again they undergo to phase 2 biotransformation. So after phase 2 biotransformation, these whatever the hydrophilic, lipophilic and highly lipophilic and polar compounds is converted into inactive form and enter into the urine. So what are the biotransformation conversions? So previously we discussed not only the active form of the drugs, the inactive form of the drugs. When these drugs enter into the body, it undergoes the metabolism in liver and it is converted into inactive form. See here, it causes the conversion of an active drug to inactive or less active compound. So in the first type of conversion, the main motto is pharmacological inactivation. So when we take any drug into the body, so it is enter into the blood circulation and it shows the mechanism of action and it will go to the liver for the metabolism. So here the active component is converted into inactive component, which we called it as pharmacological inactivation. See here the examples are the phenobarbital, which is an active moiety converted into para-hydroxyphenobarbital, and the phenytoin, which is active form converted into para-hydroxyphenytoin, and procaine, para-aminobenzoic acid, and griseofulvin, 6-D-methyl griseofulvin. So all these drug substances initially will take in the active form. So after showing mechanism of action, it is entered into the liver and these are converted into inactive hydrophilic form and excreted through urine. So the second conversion, it causes the conversion of an active moiety to more active moiety. So initially in the first thing we discussed about the active form of the drug is converted into inactive form. But here the active form of the drug substance is converted into more active form. So we called it as toxicological activation. Say for example, if any drug will take into our body. Say for example, paracetamol 500 mg for the fever will take into the body to reduce the temperature. So initially it will act on the body and it will reduce the temperature. So later on, if it is entered into the liver and it is converted into more active form, what happened? Again and again, it acts on the hypothalamus and reduce the temperature. So gradually the patient go to coma. So this is called toxicological activation of the drug. See for example, codeine, so which is converted into a morphine, which is a narcotic drug. Generally, we are used for anesthetic purpose. So for local anesthesia and general anesthesia, these codeine and malatone and halothane, so all these drugs are used. So if we use large quantities of this drug, when this drug enter into the liver, it is converted into more active form. So thereby we'll get the sedation and later on we'll get the different effects. So next, Next conversion, it causes the conversion of an inactive drug to active form. So we already discussed, so prodrug. So initially these type of drugs are in inactive form. Why these drugs are in inactive form means, so if any drug is sensitive to the external temperature, we'll mask the drug to by using different types of agents as a prodrug. So this prodrug is stable to the external environment and different pHs also. So if this type of prodrug is enter into the body, it will dissolve in our stomach, and the inactive form of the drug is enter into the blood circulation. So at the portal circulation, if it is enter into the liver, so it is converted into active form and it will show the mechanism of action. Examples are phenacetin to paracetamol, and enalapril to enalapril, pivampicillin to ampicillin, sulfosalazine to 5-aminobenzoyl acid, levodopa to dopamine. So all these drugs, so which are sensitive to the environment and different exposures of pH and temperature, so we'll convert these drugs into prodrugs. So for in some cases, to increase the solubility also, we'll convert the drug as a prodrug. So then by dissolving the drug into the blood circulation and it is entered into the liver, again it is converted into the active form and it will show the mechanism of action. So the fourth one is, it causes the conversion of an active drug to equally active metabolite. So by this type of metabolism, same active moiety is entered into the metabolism and the metabolite also equally potential to the drug. So example, digoxin to digoxin, and diazepam to nordiazepam, amitriptyline to nortriptyline. So all these types of drugs. So there's digoxin used for the congestive heart failure, and diazepam which produce the suppressant, and amitriptyline, so which is also used to reduce the excitement. All these types of drugs, when it is enter into the metabolism also, it is converted as a active form of drug. So what are the sites of the drug metabolism? So the major site of the drug metabolism is liver. Why? Why the major site of drug metabolism is liver means, the microsomal and non-microsomal enzyme systems which are used for the metabolizing of different varieties of drug by non-specific methods are present in the liver. That's why liver is the major site of the metabolism compared to other sites. The secondary organs like kidneys. In kidneys, the proximal culvated tube is also a part for the metabolism, and lungs, and the drugs which we are taking nasal route. Some drugs are metabolized in the lungs only, and testes and skin epithelial cells and intestine and brain. All these are the secondary metabolic sites for the drug metabolism. But the major, up to 80% of the drugs are metabolized in the liver. Remaining small amounts of the drugs is metabolized in the other organs. So what are the metabolic enzymes? So till now we are discussing different types of metabolisms and how the drug is converted into active to inactive forms, etc. So here, by which process the drug was converted from active form to inactive form? So those are called as enzymes which are present in our liver. So the microsomal enzymes. So these microsomal enzymes which are present in the smooth endoplasmic reticulum of liver cells. So there are different types of cell organelles in cell, right? So like nucleus, mitochondria, endoplasmic reticulum, Golgi bodies, everything. So in the smooth endoplasmic reticulum, these types of microsomal enzymes are present. These type of enzymes which are used for the phase 1 biotransformation. So which are used to convert the lipophilic drug to hydrophilic drug by the addition of a hydrophilic moiety such as OH and carboxylic groups into the drug molecule, or unmasking the hydrophilic moieties which are present in the drug. Say for example, for some drugs, because of the instability, we'll convert those drugs into salts, anything as like diclofenac, potassium, ranitidine, hydrochloride, like that. So if the drug contains the hydrophilic moiety is masked by any other means, so these microsomal enzymes used to unmask the hydrophilic moiety which are present in the drug substance. So thereby these macrosomal enzymes increase the hydrophilicity of the drug. So here this macrosomal enzymes includes the flavin monooxygenases, cytochrome P450, NADPH, cytochrome C reductase, UDP glucoronosyl transferase. All these are the examples for macrosomal enzymes. So here we called it as monooxygenases. So why? Because so for the drug, if you'd like to add the OH group, so we'll use, say for example, this is the drug RH. So we'll use oxygen molecule and NADPH and H plus molecules, and it convert the drug into hydrophilic form by adding OH group to the drug and NADP plus H2O. See here, in the molecular oxygen O2, these enzymes take one oxygen and add it to the substrate, which is nothing but our drug. So here from the molecular oxygen, only one oxygen added to the substrate, the cause of the enzymes for the addition of one oxygen group for the substrate, that's why we called it as monooxygenases. Mean to say only one oxygen in the molecular oxygen is added to the substrate which we would like to convert into a hydrophilic form. So that is called monooxygenases, and these are also called as multifunctional oxidase enzymes, because here the other oxygen molecule and this hydrogen which is present in the NADPH and hydrogen molecule convert into water molecule. So here one oxygen is attached to the substrate and water molecule was released and NADP plus was released. Here multifunctional, three types of functions were acted to form the hydrophilic drug molecule. That's why we called it as multifunctional enzymes. So the second type of enzymes are non-macrosomal enzymes. So these non-macrosomal enzymes are also present in the liver, but these enzymes are present in cytoplasm and mitochondria. So of the cell wall, okay. The special point for this non-macrosomal enzymes are these non-macrosomal enzymes are in dissolved forms. In the cytoplasm. So for this nature, the drugs which are hydrophilic form also, which are dissolved in the cells also, these non-macrosomal enzymes is used for phase 2 metabolism and convert the soluble drug substances into inactive form. So why this non-macrosomal enzymes are mainly for the phase 2 metabolism? The drugs which are already converted into hydrophilic form in phase 1 metabolism, and the drugs by nature, these drugs are hydrophilic form, means these non-macrosomal enzymes undergo the phase 2 metabolism and convert the active hydrophilic form of drugs to inactive hydrophilic form. So examples are esterases, amidases and flavone oxidases. So these are the examples. So mainly these non-macrosomal enzymes are participated in the conjugation reactions. What is the meaning for conjugation? The conjugation means covalent bond formed between drug molecule and what is the hydrophilic moiety. So these non-microsomal enzymes are used to form the covalent bond between the drug and the hydrophilic group which we would like to add to the drug molecule. See here, microsomal enzymes, a group of enzymes associated with certain particulate fraction of the liver. So these are mixed function oxidases. We already discussed in the last slide the mixed function and monooxygenases. Why we called it as mixed function and monofunction oxygenases. So by taking one oxygen molecule from the molecular oxygen, that's why we call it as monooxygenases. It performs multiple functions in the one reaction, that's why we call it as mixed function oxidases. So for these reactions we need NADPH and oxygen. And two most important microsomal enzymes like cytochrome P450 and cut P450. So these two enzymes are mainly used in the phase 2 metabolism to add the hydrophilic group of the drug molecule. So the non-microsomal enzymes, these are mainly presented in solubilized form, and these solubilized form which are used in the phase 2 metabolism to inactivate the drug molecule.
Like, share and subscribe. Hit the bell icon for more updates.